Today we're going to start Unit 9 for Bio 102, and it's going to be on immunity and health. So your body's going to have different ways to protect you against disease-causing invaders. So with this, we're going to have 3 lines of defense that help prevent and fight pathogen attacks. Many of us do not really consider pathogens as disease-causing microorganisms. You have these on your body, you find them around you on everyday things. You also don't really consider the effects that they have until you hear them talking about it actually being an illness or something's going around. For example, with COVID or with the seasonal flu or even H1N1. So microorganisms are going to be the cause of a lot of things that we consider to be these diseases or these illnesses, but we're also going to find them on our skin and on everyday things, and they actually can be good for us too. So you have the good and you have the bad. So basically with these microorganisms, they're going to be a vast majority of microscopic things that are going to be found on everyday items, and most of them we don't even consider, like I said, to cause disease. But every now and then you come across some that will cause disease. Or they could cause disease if they outgrow or overgrow in a certain situation. For example, you have normal flora or a specific type of bacteria and fungi and other things that stay on your skin at all times. And with that normal flora, as long as everything stays in check, then everything will be okay. But let's say that you're taking an antibiotic and that bacteria starts to die down because of that antibiotic. It could be to treat some other condition, but it will kill other bacteria in your system or on your skin even. And so let's say the number of bacteria that we consider normal bacteria on your skin start to die down. Then if you have fungi on your skin, then they can overgrow because they no longer have competition or anything to compete with. So you might start to see something like ringworm develop on your skin or other fungal infections. So the immune system is going to provide protection against an enormous variety of pathogens, and these can include things like bacteria, viruses, fungi, parasitic worms, and protists. So with the immune system, you're going to have several different ways that your body's going to naturally protect itself. You have physical barriers, you have non-specific immunity, and you have specific immunity. Now with these 3 main divisions, the physical barriers is going to be the first thing that it comes across. It's going to form basically an impenetrable wall and keep pathogens from entering the body and entering the tissues. And so this physical barrier, or what we talk about as the physical barriers, is going to consist of things like your skin, your mucous membranes, and different secretions that they make. Now going in with the non-specific immunity, that's going to recognize and destroy pathogens that might breach that external barrier. So they've gotten past that main first line of defense, and they're going to respond to all pathogens in the same way, and they do this usually within minutes. The third line is going to be specific immunity. And so that's going to go in and is going to destroy pathogens that are not killed by the non-specific defenses that breached their first line. They're going to recognize specific pathogens, and then they're going to form a memory of each one of those. So if they were to ever come back, they remember them and they can send out their response. So what they're going to do is they respond to the infection, usually anywhere from within hours to days. So all 3 of these are going to work together, and that way they're able to protect you against different types of diseases and different infections. So external barriers are going to prevent pathogens from entering your body. So your body's going to have ways to prevent these pathogens from entering the body, the cells, and the tissues. So the integumentary system is going to be made up of skin, sweat glands, oil glands, hair, and nails that protect you from mechanical stresses caused by external forces such as pressure, sunlight, dehydration, and, of course, pathogens. It's going to be considered the body's first line of defense. Consists mainly of, like what we've already said, the skin, mucous membranes, and oil and sweat glands. And with this main line of defense, it's going to give you 6 defense mechanisms. So looking at the first with the integumentary system, you have your hair. Hair's going to help you pick up on any type of, if there's a bug or anything on your skin crawling, it helps you pick up on that. It also helps to protect that skin from sun and UV rays. You have nails, which can help you protect. It also has the skin surface, which is going to be an impenetrable surface there. In with the hair, notice you're going to have oil glands. These oil glands are also known as sebaceous glands. They create a substance called sebum, which is going to be the oil, and it's going to have enzymes in it that help fight bacterial infections. You have sweat glands, which will also contain substances within them that fights bacteria. You have the fat cells that can be found at the bottom. That's going to help with insulation, so it helps to keep you warm, and it helps with any type of mechanical defense as far as bumping into something. It helps kind of cushion and pad. The nerve endings and nerve fibers are going to pick up on any type of sensation that needs to register with the brain that might be important. For example, it could pick up on if things are hot or cold. It picks up if things are rough or smooth, and it picks up on pressure. And then, of course, you have your blood vessels. These are going to feed the tissues as well as take away any waste products. And along with that, you're going to find the white blood cells that are traveling along in there to actually help survey what's going on. And so it acts as a surveillance system to fight any type of microorganisms that may have gotten in. One question a lot of people have is, are toilet seats dangerous? Is there a hidden danger there? Well, the good news is that human pathogens cannot survive outside of the body for very long. And so the pathogens actually need an access point. So if there were, let's say, bacteria or different types of viruses that might have gotten on that toilet seat, for the most part, they don't survive very long without contact with the human skin. And then once they make contact, lots of times they do need an entrance point. With that entrance point, there usually needs to be some type of abrasion there in order for it to enter into that person's body system. And so most of the time, those two things do not come together and actually cause any type of infection or illness. Now, with the 6 lines of defense, we talked about the 6 mechanisms. The first, of course, is going to be skin. It's going to form a nearly impenetrable barrier that keeps pathogens from entering the body. Then you're going to have lysozyme and other enzymes. Lysozymes are going to be found in saliva and tears, and then you have digestive enzymes that are going to be found in the small intestines, which will help kill a lot of that bacteria. Acidic secretions can be found in things like stomach acid, vaginal acid secretions, acidic urine, and all of these are going to be protected within that digestive system, the reproductive and urinary tracts from bacterial infections. You have cilia. These are going to be hair-like extensions on the surface of the respiratory tract that are going to move mucus and trapped pathogens up and then out of the lungs. Now you can either spit these out or sometimes people swallow them. If it is swallowed, then it simply goes into the acidic secretions that are going to be found in the stomach. So the stomach acid will destroy it. Tears are going to be a fluid that contains antiviral and antibacterial chemicals that can wash away microorganisms from around the eyes. And then you have your earwax, which is going to be a sticky substance that can trap microorganisms in the ear canal. So with the skin being that impenetrable barrier, it also is going to actively secrete chemicals that inhibit bacterial growth. They're going to have a lysozyme, which is going to be an enzyme that can kill bacteria, and it's going to do this by damaging that bacteria's cell wall. You damage the cell wall, it will end up killing the bacteria because then it can't protect itself. It's like damaging its skin. And most bacteria have a peptidoglycogen cell wall. Right, the respiratory tract is going to be protected by mucus. So in the respiratory tract, you have the hair-like extensions that are going to be found in the epithelial cells, and they are called cilia. They're going to continuously move mucus up through the system. That mucus actually entraps the pathogens as you breathe them in. So as you breathe in, it's going to actually help clean that air as well as warm and moisten it before it makes it to the lungs. So once it traps those pathogens, it helps you to cough them up, sneeze them up, and hopefully out. If you don't spit that mucus out, like I said, if it does go into the esophagus where you swallow it, the stomach acid will destroy those pathogens there. Protecting the eyes, they're also going to have like a physical wash that they can wash away these microorganisms with. Tears also will contain lysozymes, and they also have antiviral and antibacterial chemicals in them. When protecting the ears, they have the physical property of earwax that's going to trap those microorganisms in there. The slight acidity and the lysozymes that you're going to find in the earwax are also going to serve to inhibit the growth of some of those microorganisms if they find their way into that ear canal. So the nonspecific division of the immune system is going to recognize and fight pathogens while signaling for additional defenses. So your body's going to recognize a security breach anytime any of these microorganisms or pathogens make their way into the body. So this is going to be considered the body's second line of defense. With it, it's going to work with a three-step process. So when pathogens penetrate the skin or the mucous membranes, the first thing it's going to do is it's going to identify the intruder. So here, nonspecific immune cells are going to recognize molecules that are going to be found on the surface of the pathogens. Then they're going to bind to them. Once they bind to them, they're going to mark these pathogens as invaders. So now they're actually going to have a signal on them that lets the body know they do not belong there. So after they do this, they're then going to call for backup. So the immune system cells are going to secrete signaling proteins called cytokines. These are going to recruit more immune cells to that site of the infection, and then they're going to warn them to protect themselves. Next, you have attack and removal. So from here, specialized immune system cells are going to destroy, break down, and ingest both pathogens and any infected cell that was marked as being infected. So this is where one will sacrifice itself to save the rest of the cells around it. So with cellular communication, that's going to be its chief way that cells are going to be able to talk to one another, and they're going to do this by secreting a chemical which is going to be a signaling protein, and it's called a cytokine. So these cytokines are going to secrete or be secreted by one of the immune cells that can bind to a receptor on the other immune cell, and then it's going to signal them to respond in different ways. For example, they can recruit more immune cells to that site of infection. If you have what's known as an interferon, interferons are going to be an important type of cytokine. It's going to be produced by cells that are infected by a virus. And so what these are going to do is they're going to alert other cells to turn on protective measures that are going to help them resist being infected by these viruses that are around them. So it puts them on high alert. Next, we're going to have our white blood cells. With the white blood cells, they're going to be specialized cells that play roles in both specific and nonspecific immunity. They're going to be produced by the red bone marrow, and so we're going to find these at usually the ends of the bone. So we've referred to those as the epiphysis or epiphyses if we're talking about both. And so within that red bone marrow, that's where they're made, along with red blood cells, plasma, and platelets, and they're going to be released into the bloodstream, where they're then going to patrol the body's tissues and search for invaders. So there's going to be 4 different types of white blood cells. And so all of these are going to be part of your nonspecific immune system. Now the first is going to be a neutrophil. Neutrophils are going to be phagocytic cells. They're going to ingest small organisms and primarily bacteria. They destroy both pathogens and themselves, and they're going to be the first to arrive on the scene of an infection site. Next, you're going to have your macrophages. These are going to be phagocytic cells that ingest whole pathogens, and they also ingest large debris at sites of injury. They're then going to present pieces of that pathogen on the surface of the cell, and that's going to help to advertise that there's an infection inside of the body so the specific immune system can come in and help. Third, you have your dendritic cells. These are going to be phagocytic cells that present ingested pathogens to cells and specific immune systems. And then last, you have what's referred to as the natural killer cells, or sometimes you might see them referred to as the NKs. These are going to kill body cells that are infected by pathogens by poking holes in their cell membrane. When they poke a hole in the cell membrane, it will cause that cell to lyse or to explode. They're going to play a role also in recognizing and killing cancer cells. So they're constantly patrolling through your system, and they're looking for cells that aren't acting right or proper. And they will notice when a cell quits doing its job and simply continues to go through mitosis. And so the natural killers then will be the cells that take those cancerous tumor-causing cells out. So we've talked about phagocytic cells. These are going to be cells that are part of the white blood cells that can engulf, ingest, kill, and break down foreign pathogens and particles. With the three types of white blood cells, the neutrophils, the macrophages, and the dendritic cells, all of these types are going to act as phagocytic cells. And so with these, the dendritic cells are also going to have an important role in stimulating the specific immune response. The neutrophils are going to be circulating in the blood, and they exit the blood vessels at sites of injury and infection in tissues, so anywhere that there's damage within the body or on the outside of the body. They're going to be able to ingest small organisms. Mainly they're looking for things like bacteria. They actually will produce a hydrogen peroxide and a bleach that they're able to dump on to this bacteria in order to kill them. Pus is going to be found in the process of killing. Neutrophils basically destroy themselves and become major parts of pus. They create a thick yellowish fluid that we often notice at the site of an infection. Antiseptics are going to be a solution that's going to be used on a body's surface to kill or to discourage the growth of microorganisms. These can be things like rubbing alcohol or hydrogen peroxide. The thing about antiseptics is that it will kill and reduce the number of pathogens, but it doesn't kill everything. If it could kill all the living pathogens on there, it's also going to kill the living tissue. So you need something that's going to be safe enough to use on living tissue, but will kill microorganisms that aren't part of the body system. So that's going to be your difference between an antiseptic and a disinfectant. Macrophages are going to be large blood cells that reside and control the tissues. They're going to engulf and digest whole pathogens, as well as any debris that may remain after the neutrophils have done their job. Next, we have the dendritic cells. These dendritic cells are going to be phagocytic again. They're going to be part of that non-specific and part of the specific division of the immune system. They're going to work as presenting cells, so they'll take parts of the foreign matter, for example, it could be a virus or it could be a bacteria, and they'll present that on the outside of the cell membrane of the infected cell. They're also going to migrate to the lymph nodes. And when they do this, that's going to be populated by cells with specific immunity. And so that's going to go in for the second round of attack, or a more specific attack on whatever organism is actually infecting that person. The fourth type of white blood cells, like we said, were the natural killers. Remember, these are the ones that are constantly in your bloodstream that are searching for things that don't belong, things such as bacteria and other pathogens, but also they're surveilling its own body tissues. They're looking for cells that aren't doing what they're supposed to do, and these are going to be your cancerous cells. So remember, these are the ones going around poking holes in the cell membranes and destroying the cells that either are infected or that are cancerous. So with the natural killer cells, they provide first line of resistance to viruses, like we said, by poking holes in their cell membranes of those infected cells, and they also kill the virus that's inside of those cells. Complement proteins are going to be a nonspecific system that can also quickly recognize and destroy invaders thanks to some circulating defensive proteins, and these collectively are going to be referred to as complement proteins. So looking over here, you see them recognize pathogens. They help fight them in various ways. They will stick to them and increase the ability of a phagocyte to consume them by directly destroy, or they could directly destroy them, like is seen here. So with some of the nonspecific systems to respond, you have what's referred to as the inflammatory response. So the inflammatory response is going to be a combination of events that leads to the recruitment of phagocytes and other immune cells to assist with pathogen destruction, followed by tissue healing. So with the inflammatory response, you have invading pathogens from an invading body and skin surface. They're quickly going to be engulfed by the macrophages that reside in all tissues. Those macrophages then will release these cytokines to recruit more phagocytic cells and 2 other types of white blood cells, which will initiate inflammation. Basophils and mast cells are going to trigger what's known as a histamine release. Basophils are going to circulate in the blood, and mast cells are going to be found in the tissues. So that's going to be the difference here. Both of these will trigger the inflammatory reaction by releasing histamine. You probably recognize that from allergies and allergy season. Most of the time when you get a runny nose and you sneeze a lot, you will take an antihistamine, and that is to stop these cells from releasing histamine. Histamines are responsible for causing an inflammatory response where it's going to cause the body to create more mucus and try to help flush pathogens or what is perceived to be a pathogen. In the case of allergies, usually it's some type of pollen, and it causes them to try to flush that out of the system. And so you'll take an antihistamine to stop these from doing that. All right. So looking here, the inflammatory response, so you see someone's going to cut their finger here. So after you cut your finger, let's say with a knife, invading pathogens from that knife and the skin surface are going to enter the body. Macrophages that are residing in the tissues that surround the cut begin to engulf the pathogens, releasing cytokines. They're recruiting more phagocytes and other white blood cells that will come to the area. Next, you're going to have the basophils, which circulate in the blood, and then the mast cells, which are already found in the tissues, that trigger inflammatory reaction by releasing histamines. Histamines are going to cause nearby non-injured blood vessels to dilate. It's going to increase blood flow to that area, and it's going to allow a greater supply of defense molecules and cells that can fight that infection. Histamines also going to cause the blood vessels to become more leaky. That allows more neutrophils to exit the bloodstream and to enter that infection site. And last, the inflammatory response is going to continue until the pathogens have been eliminated from the skin and it grows back, forming an impenetrable barrier once again. So with that inflammatory response basically is going to initiate specific things. It's going to cause redness, heat, swelling, and pain, but ultimately it's going to lead the tissue to a more healing state. So with the histamine reaction, like we said, we're going to have an increase in blood supply at the site of the injury, and that's going to cause the redness and heat that's associated with inflammation. Increased leakiness of blood vessels is going to make it easier for things such as neutrophils to exit the bloodstream and the mast cells that are in the tissue, so they can actually help start to destroy those invading pathogens. So with that, pain is going to be caused lots of times. So what this is going to be caused by is the macrophages and the neutrophils begin an immediate response to destroy those pathogens and release cytokines. Some of the cytokines are going to be released from the macrophages, neutrophils, and mast cells. And what they're going to do is they're going to activate neurons. And when they activate the neurons doing this, that's actually going to cause the pain response. So the little nerve endings are going to be triggered. And that's going to send a sense of pain to the brain. Complement proteins, you have activated complement proteins at the site of infection that cause mast cells to release the histamine, and further they're going to amplify the inflammatory response by attracting additional phagocytes. Lastly, once everything has come together and it's getting ready to heal, it's forming that scab. That scab's going to be a temporary cover that's going to cover that wound, kind of like a natural Band-Aid for the body, and it's going to prevent more pathogens from entering into that area. At the end of the inflammatory process, you're going to have collagen fibers that are secreted by nearby cells that are going to end up closing the wound and making a more stable healing area. And then eventually, once the skin underneath is healed, that scab will fall off. Fever. Fever is going to be defined as a body temperature that's going to be above 37 degrees Celsius, or what we consider to be 98.6 degrees Fahrenheit. It's actually going to be caused by the cytokines. It's got 5 functions. So 1, it's going to enhance the effect of the interference. 2, it's going to inhibit the growth of some of the microorganisms because they cannot grow in that high temperature. A third thing it does is that it enhances the performance of the phagocytes, so they actually like that warmer temperature. 4th, it's going to enhance the activity of cells of the specific immune response. And 5th thing it's going to do is it's going to enhance the process of tissue repair. Specific immunity is going to develop after exposure to pathogens. So specific immunity is going to form memory of specific pathogens. So it's going to have the recognition of more specific, so it's going to definitely know what's coming in. The different types of cells and molecular weapons are going to be used. And there's going to be a trade-off between the speed of non-specific response and a slower but much more precise response with specific immunity. So the specific immunity system is going to form a memory so the body can respond, like we said, to specific pathogens. It occurs more quickly in the future once it encounters it. So it does take a while for it to create this response, but it does keep small amounts constantly circulating in the blood so that when it comes across this pathogen again, it already knows it, it alerts the system, and it's able to make more very quickly. But at first, it does take a little while for it to create a way to defend itself with a specific immunity. So there's going to be 2 ways that you can acquire immunity. Immunity basically is going to be defined as a state of long-term protection against a specific pathogen. So one way to have this is that you can first become sick with the disease, and then your body will create or produce cells necessary to rapidly respond to these pathogens. Or you can be inoculated with a vaccine. So a vaccine is going to be a weakened or a harmless version of the pathogen. It's going to allow the body to form a memory without risk of symptoms, and then the body's going to produce cells that can then rapidly respond to that pathogen. So like we said, a vaccine's a weakened or harmless version. Lots of times it could even be a version that is no longer alive. And so once you inject it into the body does form specific antibodies for it by looking at the antigens that are on that virus. And so next time it enters the body, it's going to be able to recognize those antigens and send out an antibody. So basically what they do is they trick the body into thinking that it has the full-blown disease, and that helps it mount its specific immune response which is the antibody. So like we were talking about antigens, an antigen is going to be any molecule that induces a specific response. So the body's going to respond to antigens by making antibodies. And so those antibodies are then going to be circulating throughout the bloodstream and throughout the body. And these basically are going to be proteins that are there for recognition. They only stick to specific antigens. So it's like a lock and key system, and it has to be very, very precise. So with these, they're going to provide protection by enhancing that non-specific system's ability to recognize and destroy any type of bacteria or virus that's entered the body that could infect it. Notice how it has that specific shape to it. It will have a Y shape. It's going to be made up of 4 polypeptide chains that are going to be hooked together. And so looking here, you can see different antigens that are going to be found on the outside of the influenza virus. And so a specific antibody would actually be made for each one of those. And so like we were looking at this Y shape here, it's going to consist of 4 polypeptide chains that are joined together. You're going to have 2 long. These are going to be called your heavy chains. And so they're going to make up the main long part of the Y. And then you're going to have 2 short chains, which are called light chains. And those are going to be the 2 tops of the Y. And so that's going to be your 4 chains that you see there. And so looking here, the reason that you have to get a flu shot every year is because if you notice over time, these antigens or these markers that are going to be found on the outside of this flu cell or this virus are going to change. And so as they change slightly over time, that is going to cause them to not respond to the same antibody. So the body's going to have to constantly be making new antibodies for the more recent version. Now, the reason that they change is because most of these viruses like this that change a lot are going to be referred to as RNA viruses. RNA viruses don't have the same editing or the same proofreading that DNA does. And so when it copies something, if it makes a mistake, lots of times it does not go back and check and correct that mistake. It allows that mistake to continue on. Now, with that mistake, that will then change the overall makeup or the DNA of that virus. And so with the change of that virus, it will change the proteins and the markers. So as these change over time, the body will need to make new antibodies that will be able to actually fit onto it. So you have that Y shape, you have the long chain, you have the short chain. It's going to have 2 main regions. First region it's going to have is referred to as the variable region. That's going to be that top portion of the Y-shaped antibody. And so with that, that's going to vary from antibody to antibody. So that's what's going to help make it unique in order to hook on to those virus particles that you see. And so it's going to contain an antigen binding site that's going to be a unique 3-dimensional shape that only fits that specific virus. Now, as it changes over the years, you're going to have to make new antibodies that will change with it. So it has to fit together perfectly like a lock and key system. The next is going to be the constant region, and that's going to be those 2 long heavy chains that make up the long part of the Y. So that stays constant and it doesn't change. You're also going to have 4 main functions of antibodies. The first function is going to be phagocytic signaling. So they're going to bind to antigens on surfaces of pathogens, and they make the pathogen easier to find by the phagocytes. Second, you're going to have antigen clumping. This is where antibodies make the pathogens and free-floating antigens clump together. And so it's going to make the pathogens easier to find by the phagocytes. The third thing it does is it prevents the cell, it is a prevention of cell entry. So basically antibodies are going to coat the surface of these pathogens, and it prevents them from being able to enter the body cell. And then the fourth thing that it does, it has complement protein signaling. And so they're going to recruit complement proteins that are going to come along and poke holes in the pathogen membrane and cause it to burst. So every year you need to get a flu shot because it has changed over time. You're going to have to be able to create those new antibodies. And that basically is kind of like what we consider to be making you a moving target. So just like influenza virus, the rhinovirus, which is going to be the nose virus or the snotty virus that people tend to get about once or twice a year, is going to be one of these. It's one that changes constantly. So you know that that is going to be a RNA-based virus. So each time you catch the cold, you're reacting to a different version of that same virus. It's an entirely, or it could be an entirely new virus. So it could have been a mutated version, or it could have been a version that you hadn't had before. There's going to be approximately about 200 different viruses that produce cold-like symptoms. So it's hard to tell exactly which one you have sometimes. So each time you catch the cold, you're going to be reacting either to a different version of the same virus, or it could be an entirely new virus. So lymphocytes are going to fight pathogens on two fronts. Lymphocytes basically are going to be the white blood cells that are responsible for the specific immunity response. They're going to be found in two areas. They can be found circulating in the blood and lymphatic system, or they can be found residing in the lymph nodes and in the spleen. They're going to have antigen receptors, and so antigen receptors are basically going to be proteins that are going to be found on lymphocytes' plasma membrane. They're going to stick out from the cell surface, and then they can bind to specific antigens. So each lymphocyte can bind to just one type of antigen, kind of like the antibody. There's going to be two major types of lymphocytes that you have in your body. You can have the B lymphocytes, often referred to as B cells. These are going to arise and mature in the red bone marrow. They're going to combat pathogens by releasing antibodies into the body fluids when antigens are detected. So the B cells are going to be the ones that make antibodies. Know that these are the ones responsible for antibody production. The next type is going to be a T lymphocyte or T cells. These are also going to be produced in the red bone marrow, but they're going to be sent to the thymus in order to mature. And so they have to go through immunocompetent training. Basically, they have to learn what tissues that they can attack and what tissues they cannot. It's hard to believe that only 2% of your T lymphocytes actually make it through this process. 98% are destroyed, and the products that they're made of are just basically absorbed and reused by the body. So like we said, they're produced in the red bone marrow, but they're sent to that thymus to become immunocompetent. Once they're immunocompetent, they're going to combat pathogens by directly destroying infected cells. And they have two major types. You have what's called a cytotoxic T cell, and that's going to directly kill infected cells. And then you have the helper T cell, and that one's going to help regulate the activity of B cells and cytotoxic T cells. It causes the B cells to produce antibodies, and it causes the T cells to kill infected cells. And so with these, they're going to fight pathogens on two fronts. First, you have what's referred to as the humoral response, and then you have the cell-mediated immunity response. And so with the humoral response, it's going to provide protection against specific pathogens and toxins that are found in body fluids. It's going to secrete antibodies into those body fluids, and it's going to make pathogens easier to engulf and destroy phagocytes. With the cell-mediated immunity, it's going to provide protection against specific pathogens and toxins that are found within the body cells. It's going to be carried out by the T cells, and it will directly destroy invading pathogens and infected cells. And so with lymphocyte specificity, each lymphocyte's going to have only one type of receptor that recognizes only one antigen, and it has many copies of this receptor on its surface. So to defeat the specific pathogen, numerous identical lymphocytes that are going to have the same antigen receptor are going to be needed. All right, clonal selection is going to help in fighting infection now and later. And so when the lymphocyte does come into contact with the antigen specific to its receptor, a sequence of events is going to begin to happen, and it doesn't end until that antigen is going to be destroyed. So first thing that happens, it recognizes it. Recognition occurs when it, like we said, bumps into it and comes in contact with it. Next, it's going to have clonal selection. And so when that B or T cell binds to the antigen, that cell and its descendants are going to divide many, many times to create clones of themselves with that same antigen on it. This process is known as clonal selection. It's cloning itself. That's going to ensure that there's going to be enough B and T cells to recognize and respond to that specific antigen or pathogen that's going to be there. And so here you see recognition, you have your clonal selection. Next, you're going to have the effector cells coming in and attacking. So the cells recognize the antigen and immediately take action, leading to the destruction of that antigen. And then fourth, the memory cells are going to remember it. They're going to remember the antigen in case the body's infected with the same antigen in the future, and it's going to help it have a faster, stronger secondary response that can come to the body's aid. So with those effector cells, those are going to be the responders. They're the ones that come in to help. Plasma cells are going to be derived from B cells. They're the effector cells in the humoral response because they secrete antibodies. With the primary response, that immune system's first interaction with the pathogen is going to be there, and the effector cells are not immediately going to be available. The cells of the primary immune response must be first generated through clonal selection. Now, for it to be able to do this, it could take up to 2 weeks to produce effective numbers. So this is why the first time when you're exposed, it's going to take you a little while before you fight it off. But then the next time that it recognizes it, it's able to create more of these clonal responses, and it can send in a reinforcement or an army right away to destroy it. The second type of lymphocytes are going to be produced through clonal selection like the effector cells, and they remember the antigen. So if the body is going to be infected with the same antigen, it's ready to attack it. So here's a visual showing the longer period that it takes once the body's exposed before it can make its first response. And then by the second response, you see within time that secondary response will kick in, but it will be able to come to the body's aid a lot sooner in any of the subsequent infections after that. Right, so the structure of the antibody, this is what we've talked about already. We had the two short regions, which are referred to as a variable region. These are going to be your short chains or light chains. Then you had the long chain, and that's going to be the chain that's going to be the constant chain. It's a variable region. That's going to be the top of the Y. It varies from antibody to antibody. That's going to be the one that is specific to the antigen. And it's a three-dimensional shape, and it's going to fit together with that antigen like a lock and key system. The constant region stays constant in all antibodies. That's going to be the long body part of the Y. So how do antibodies work? Well, they're going to use their nonspecific cells of the nonspecific immune system, such as macrophages and neutrophils, to kill that pathogen. So the antibodies are going to act as beacons, like we said before, and they're going to attract these phagocytes to these cells in order to have them devoured and destroyed. So antibodies act in two other important ways. They can cause pathogens or antigens to clump together, like we said, or they can coat the surface so they're not able to enter other cells. So this here shows you phagocytic signaling. It shows how it could cause antigen clumping, prevention of cell entry, and has the complement protein signaling. Passive immunity is going to be the transferring of antibodies with a given specificity and it's going to be termed as passive immunity. So during pregnancy, antibodies are going to be produced by the mother, and they're going to be transferred to the baby's bloodstream to protect that baby for the first few months of its life. Also, this passive immunity can be transferred through things like breastfeeding, so it can come through the breast milk. Any antibody that the mother carries is going to be transferred from the breast milk to the baby, and then the baby can use those antibodies as long as it's feeding. Cytotoxic T cells and helper T cells are going to serve different functions. So what happens to all the body's cells that already have a virus hiding inside of them? Well, it's going to be the role of that T cell mediated response to fight pathogens that are already inside of cells. So the cytotoxic T cell has the effectors of the cell that are going to be in a mediated response. They directly kill the cells that are infected with that pathogen. The helper T cells don't directly kill infected cells, but they stimulate other immune cells to come in. So it's going to be required in the stimulation of things such as B cells to produce antibodies and the cytotoxic T cells that will come along and then kill the infected cells. And so what happens there is like the others, you have presentation. With the presentation, an antigen-presenting cell is going to display digested particles of the virus to the helper T cell, and it's going to recognize that virus, and it's going to be able to come in and activate. With activation, it's going to bind to the antigen-presenting cell and activate the helper T cell, which then produces cytokines that are going to activate cytotoxic T cells as well as B cells in the humoral response. Then you have clonal expansion, where both the helper T and cytotoxic T are going to undergo basically a cloning, where it makes many, many more of them. Then you have maturation. Other cytokines are going to be produced by the helper T cells and make the cytotoxic T cells mature and ready to fight the pathogen at the site of the infection. And so it helps mature them there. Then they go in and they destroy that pathogen there. So how do cytotoxic effector cells fight the internal pathogen? Well, they kill the infected cell, and then they're going to use the proteins of that destroyed cell membrane or promote apoptosis. And apoptosis, remember, is going to be a programmed cell death or cell suicide. Malfunction of the immune system can cause disease. Autoimmune diseases can occur when the body turns against its own tissue. So basically, autoimmunity occurs when an individual's immune system responds inappropriately to an individual's own cells and tissues as if they are pathogens. So it can't recognize if tissues are going to be itself or non-self. And so it attacks everything, even the self cells. Lymphocyte receptors wrongly recognize the individual's own molecules in the cell structure as antigens, and so the humoral and or cell-mediated immune responses can be initiated by the autoantigens. And so there's going to be three types or three examples of autoimmune diseases. First, we have type 1 diabetes. And here you see with type 1 diabetes, the T cell receptors incorrectly recognize healthy pancreatic cells as antigens. The cytotoxic T cells will then destroy that person's pancreatic cells. And as the pancreatic cells are being destroyed, an affected individual can't make insulin anymore. So they're not going to be able to use that insulin to get the sugar out of the bloodstream and to feed the tissues. Another autoimmune disease is multiple sclerosis. With multiple sclerosis, it's going to be a disorder in which myelin that surrounds nerve fibers of the brain and spinal cord are going to be under immune system attack. And this is going to occur actually more in women than in men. Individuals will begin to exhibit symptoms between the ages usually of 20 and 40 years. So it's a long period, but that's the time when we usually start to see it show itself. Symptoms can include blurred vision, arm and leg weakness, trouble with balance, and tingling sensations. Over time, people will eventually lose their ability to use their muscles. They'll lose their ability to walk and to do things that they normally do on a day-to-day basis. A third type would be like rheumatoid arthritis. This is a disorder in which the joint linings are going to be affected, and it leads to pain and swelling. This also occurs in women more than men, and individuals begin to exhibit symptoms usually between the ages of 40 and 60, sometimes earlier. AIDS is going to be an immune deficiency disease. The human immunodeficiency virus, HIV, is going to be the virus that causes AIDS. AIDS is going to be once it reaches its final stages, and it stands for Acquired Immune Deficiency Syndrome. It's going to infect the immune system cells, including helper T cells and cells that are going to be crucial to survival. So with HIV, it's going to be an RNA virus, so that means it's going to mutate fast. It is going to be able to go in and compromise that immune system, like we said, by attacking the T cells. It's going to leave individuals susceptible to infections and different types of cancers. Also, viral infections can occur as a result of contact with an infected individual. And it will go through 3 phases and 7 steps. So first you have what's known as the acute phase. This is the HIV infection. So what happens is HIV infects the helper T cells, macrophages, and dendritic cells, and the humoral and cell-mediated immune responses are going to be eliminated over 99% of that virus. Then you would go into a chronic phase, and that's when the person is infected but not showing symptoms. So the virus doesn't destroy the primary response. It just kind of lies dormant inside the infected helper T cells, or it's replicating and infecting new helper T cells, but you don't know yet. The infected helper T cells are then going to be destroyed by viral infection or by the immune system's normal responses. And then you're going to have those new helper T cells come around, but the new virus infects them, and eventually the virus is going to increase in number and the helper T cells will decrease. And so here you see that acute phase and you see that chronic phase. The third phase is going to be full-blown AIDS. This is when you're actually in the final stages. And that one's going to occur in 2 steps. So without the helper T cells, B cells, and cytotoxic T cells, they cannot be activated. Common pathogens that are normally kept at bay by healthy individuals can actually lead to serious illness, if not death. So like we said, HIV is going to be an RNA-containing virus. Since we know it's an RNA-containing virus, that means it's going to be able to mutate very rapidly, so the body's going to have a hard time keeping up with it. This is also one of the reasons that they have a hard time trying to treat it. RNA viruses are very hard to treat because they're constantly changing. We even see that with COVID. COVID is an RNA virus. So within a single host with HIV, that virus's surface proteins can change with each new round of replication. So if a drug or some type of treatment is working, it could work for a matter of months. It could work for years. Or it could work for a matter of months or weeks and all of a sudden not have any effect on it. So current therapies for HIV infection have been successful at increasing the length and quality of life for those infected. The regime is going to be called a combination of therapy and includes numerous drugs that affect HIV's ability to replicate or infect new cells. And so like I said, it might work for a while. Certain ones might work for a while. They constantly have to go and reconfigure different treatments for people. So drugs keep the virus number low and maintain a suitable number of helper T cells. But HIV resistance to the drugs is a fear shared by both patients and clinicians because we do see this. HIV around the world, if you look at this map here, you see that the darker reds, the deep maroons, those are going to be the areas that have the highest infection rates. As the United States, you see that we have about a 0.5 to 0.9% infection rate. You see parts of Russia, and then sub-Saharan Africa and southern Africa are going to be parts that are going to be very highly infected, along with some parts of South America and Central America. Allergies. These are going to be inappropriate immune responses to a harmless substance. So the CDC, or Center for Disease Control and Prevention, estimates that approximately 3 million children in the United States have some type of food allergy. 90% of that's going to be caused by just 8 foods, and that's going to be things such as peanuts, tree nuts, milk, eggs, fish, shellfish, soy, and wheat. So allergies are going to be, like we said, an inappropriate immune response to what we should have as a harmless substance. Basically, the allergens are going to be these antigens that cause the allergic response. Remember, the antigens are going to be things such as pollen that we shouldn't be allergic to, but the body's picking up as it being something that we are allergic to. Or it could be an inappropriate response to an antigen that's on food. And so with this, it's going to have 2 responses. The first response is going to have 2 steps. The body first encounters the allergen, and the humoral response is going to occur. Memory cells are going to form, and plasma cells secrete antibodies specific to that allergen. Second, you're going to have your mast cells come in and become sensitized, and the allergen after antibodies bind to the surface. The second response is also going to have 2 steps. The allergens are going to bind to antibodies still attached to mast cells, causing those mast cells to release histamine. That histamine is going to cause blood vessels to dilate and become leaky, causing inflammation. And so most allergic reactions occur in the digestive or respiratory tracts because this is where the body's first going to encounter allergens that are either going to be eaten or inhaled. So treatments, you can take an antihistamine to alleviate some of these allergies, as these medicines are going to block that inflammatory effect of the histamine. Other medicines are going to be able to block the mast cells from releasing histamine, and sometimes you can take steroids to block the production of cytokines from that immune cell. Anaphylactic shock is going to occur in an individual with severe allergic reactions. So it's going to cause severe respiratory distress. So basically what's going to happen there is the throat's going to swell and asthma is going to develop. It's going to cause low blood pressure and result in tissue swelling. So it can completely cut off the air supply to that person. You can have skin allergy testing that's done in this process. Microscopic amounts of 1 or more potential allergens are going to be injected under the skin. They're basically going to put a grid or a map across the skin so they can keep up with what they're injecting or putting on the skin to see what ends up showing the reaction. Asthma, unfortunately, is going to be, we see, is on a rise. Asthma results in an airway that's going to be swollen and inflamed and leads to chest tightness, coughing, wheezing, and breathing difficulty. There are both going to be genetic and environmental factors associated with asthma. The CDC estimated that childhood asthma more than doubled from 1980 to the mid-1990s. As of now, there's no clear explanation why this increase is happening, but they think it's because of a lot of the pollutants that are going to be in the air. That's going to finish us up for Unit 9.